Good morning. I'm Jonathan Saltzman, a biotechnology reporter for the Boston Globe. And today we're discussing a medical milestone, the recent approval of the first drug to use the revolutionary gene editing tool called CRISPR-Cas9. In December, the FDA approved the medicine, which is marketed as casgevy by Vertex Pharmaceuticals and CRISPR Therapeutics, for sickle cell disease. That's a blood disorder that causes crippling pain and, according to experts, didn't get the attention it deserved for many years. The drug, by the way, has a huge price tag, $2.2 million per patient. We have three guests today. After I pose some questions, viewers can ask their own by using the Q&A toolbar on the bottom of the screen. First, we have Marie Tournier, a recent graduate of Cornell University who received the treatment in a clinical trial in 2021. We also have Dr. Stuart Orkin, a physician researcher at Boston Children's Hospital and Dana-Farber Cancer Institute. He helped identify the gene that casgevy SNPs to treat sickle cell. And finally, we have Dr. George Daly, the Dean of Harvard Medical School, who is a hematologist and has treated patients with sickle cell disease. Welcome to all of you. I'd like to start with Marie Tournier. You underwent this treatment when it was still experimental at Children's Hospital of Philadelphia. What prompted you to participate in this trial?
So before having transplant, I was someone who constantly was in pain. I think by high school, I was having crises every other week, and I was in the hospital every few weeks. And it was just so unsustainable. I couldn't do anything. I couldn't live the life that I wanted to lead. So when I heard about transplant, it was a new hope for me. And I was very excited about the prospect of being cured.
Now, Marie, you said transplant, and for our viewers who aren't that familiar with CRISPR, um, there's a transplant of gene edited cells. Isn't that correct?
Yes.
How does that process, can you briefly describe how that process works and how long it takes?
Yeah, so for me, it started my sophomore year of college. And first steps were to undergo apheresis. And that's the transferring of blood cells to essentially have new fresh blood in your system. Um, and then we collected cells in order for them to be edited. And then after that, I was hospitalized for the actual transplant itself. I had to undergo chemo, and that process itself took, I think, 8 weeks. So from the beginning of apheresis to the actual hospitalization, it was about a year that it took.
Right. And so, how did you feel after that year? I mean, chemotherapy is a grueling regimen, as we all know. How did you feel? When did you start feeling better? And how do you feel today?
Um, it took a little bit for me to start feeling better. After chemo, I didn't feel like myself. I didn't want to eat. I didn't want to do much of anything really. But I'd say 4 to 6 months afterwards, I started to feel like myself. Um, and I'd say a full year afterwards, I could actually see the benefits of having the transplant. I didn't have any sickle pain anymore. I didn't have any crisis, and it was astounding. Going from having such crippling pain every single day to having no sickle pain was a complete 180.
Wow. Dr. Orkin, CRISPR-Cas9 was first described in a 2012 paper by two future Nobel Prize winners. But you made a discovery before that that would prove critical to the development of Casgevy. What was that?
Thank you. We had been working for quite some time at trying to understand how the hemoglobin protein is actually regulated, how it's expressed. And we've known for decades, actually, that there's a fetal form which is expressed while we're developing in the uterus and fetus, and that can replace sickle protein and is perfectly functional in adults. And from studies many years ago, we knew that those individuals who are fortunate to have more more fetal hemoglobin, have a less severe sickle cell disease. And so we've been studying for many years, how does one regulate this switch from the fetal hemoglobin to the adult hemoglobin? And in 2008, we had identified a regulatory factor in cells that we believed was the silencer, the thing that actually keeps the fetal hemoglobin off. And a couple of years after that, we showed that if one downregulated or removed that factor in cells, we could express lots of fetal hemoglobin, and it would actually cure a mouse that had been engineered to have sickle cell disease. So it became clear that if we could manipulate the gene in some way, we might have a potential treatment. And fortuitously, as work went on, we found a very small region, actually one base, essentially, one letter in the DNA of 3 billion bases. If we just cut there with Cas9, which is the enzyme that cuts DNA, that was sufficient to basically cripple expression of the factor and let fetal hemoglobin be expressed. And that's exactly what the therapy is. It's basically using CRISPR-Cas9 to bring that enzyme, that cutter, to this one letter in the DNA, cut it, disable B11A, and let fetal hemoglobin be expressed. And so
And, and,
that
and
was actually basic work that led to this clinical application.
And as background, and correct me if I'm wrong, Dr. Orkin, sickle cell affects hemoglobin, which is the oxygen carrying protein in red blood cells, and it
Right.
causes those round, flexible cells to deform into a sickle shape and stick to vessel walls. That deprives tissues of oxygen and causes crushing pain. But fetal hemoglobin resists that. And so scientists wanted to figure out a way to turn back that fetal globin back on. Is that correct?
It's correct. And actually, it all stemmed from a remarkable clinical description in 1949 by a pediatrician who recognized that children who were born and would clearly have sickle cell disease as they got older were not affected at birth in the first few months. And she surmised, and she was correct, that fetal hemoglobin is protective. And what it does is it replaces the sickle hemoglobin, but more importantly, it actually blocks this, what's called sickling, this polymerization or the chains that form that disrupt the cells.
Now,
This was actually the idea, the germ of the idea is 75 years old.
wow. Casgevy was remarkably effective in clinical trials, completely relieving 29 of 30 sickle cell patients of debilitating pain episodes for at least 1 year among folks who participated and were followed for at least 18 months. Dr. Orkin, does that mean these patients were effectively cured?
I think one's always hesitant to say cured, but I think it's very likely to be the case. Uh, I would expect that they would remain well for a very long time.
Dr. Daly, you're a pediatric hematologist and you treated many kids with sickle cell disease. What was that experience like?
Hmm. Well, Dr. Orkin and I are both pediatric hematologists, and indeed, we both treated many patients in our past. Both of us are some years removed from our clinical experiences, but I can tell you that any experience caring for a sickle patient is just deeply, deeply ingrained in one's mind and experience. Marie was really remarkably stoic in describing the painful crises. As a clinician, this is one of the most distressing conditions that you witness just because of the excruciating pain. Usually it's young kids or young adults who are brought into the hospital in these painful crises, um, think about it. I mean, the sludging of the blood in the tissues deprives muscles and bones of the oxygen they need. And that painful burning sensation, which some people will remember from excessive exercising, the muscle burn. But usually, if you stop exercising, it goes away in a few minutes. If you're in the setting of a sickle crisis, that pain and that excruciating suffering can last for days. And the problem is that we really, as physicians, up until recently, didn't have a lot of tools to solve this problem. We could give supportive care. We could typically give a lot of fluids. We could give transfusions of healthy blood, but mostly we would give very high doses of narcotics that would excessively sedate the patient. And I think one of the more challenging aspects of being a doctor trying to relieve suffering is when, even in the face of all of that, the patient is still suffering the pain. And that's what has been really just gut wrenching for generations of doctors who've had to care for sickle patients. But again, Marie has described the fact that her painful crises were happening frequently. She was in and out of the hospital. Think of how disruptive that is for a young kid's education. Marie was able to triumph over remarkable challenges and to become a graduate of Cornell. I mean, it's really, truly spectacular. But to then have this I mean, of course, we always worry about using the word cure, but we do believe, based on the basic science that Dr. Orkin has brought forth, that these genes have actually been corrected in Marie's stem cells, and these cells should supply healthy blood throughout the course of her life. So it's really an extraordinary revolution. It is a miracle medicine.
Hmm. Dr. Daly, sickle cell, as I understand it, was the first human disorder understood on a molecular level. Its underpinnings were explained in a landmark paper in 1949 by a future two-time Nobel laureate. And yet, for years, it didn't get the attention that other rare diseases received. Why was that?
Well, one can look at the fact that sickle cell anemia is subject to some great health and research disparities. Um, Sickle cell as a gene defect really originated in parts of sub-Saharan Africa and the Middle East in the setting of malaria. It's a fascinating really experiment in evolution that the appearance of this random mutation, which would otherwise cause such a debilitating disease, was maintained in human populations because it conferred a degree of resistance to malaria. So most of the individuals worldwide who are impacted by sickle cell anemia either live in sub-Saharan Africa currently or trace their origins to Africa. So African Americans carry the burden of this. And we appreciate and know, uh, unfortunately, that diseases affecting African Americans are often understudied. And indeed, this disease, sickle cell anemia, was subject to really a significant amount of neglect, if you will, for many decades. And I think it's in part that there wasn't strong voices of advocacy that were listened to. But of course, about in the last 10 or 15 years, especially as the advent of CRISPR, we really appreciated in the scientific and medical community that this was a disease that we could cure. This was a health disparity that we could address. And so I think it's not just a great triumph of modern science, it really proves to be a triumph of addressing a long neglected disease.
Now, obviously, none of our guests works for the drug companies that actually made casgevy. But I do think that a lot of our viewers will wonder why this drug is so expensive, like a lot of rare disease drugs. I mean, this drug costs $2.2 million per patient. It is covered, as I understand it, by insurance. Um, why does it cost so much? Dr. Orkin, do you have a perspective on that?
Yeah, I think it's always hard to evaluate price and value of medicines, but there are many medicines that are expensive. For example, chemotherapy drugs for cancer, where you might pay a large amount, but not have a cure. You might, live just a few months longer. So, I think I would actually say the price is a bargain when you consider the medical cost of a life of treating patient who had sickle cell disease. Plus, I think just the burden on the family, the problems with employment and frequent disease. So, I think it's maybe a fact of what it takes to make a drug. Making a drug is expensive. The trials were very expensive, as I understand it, to execute, and I think the price is actually a pretty reasonable one for the current therapy in the U.S. at least. So,
And
it's expensive. I hope it's covered by insurance. I think it'll be a benefit to society if it is for those patients who are cured. But we'd all like it to be cheaper, but I think this is just a fact of current medicine.
and in fact, I should point out that the independent Boston-based drug pricing watchdog group, the Institute for Clinical and Economic Review, said that as expensive as casgevy is, it could be justified given how much sickle cell disease costs to treat over a lifetime. I mean, Marie, I remember when I interviewed you a few months ago, you said, I mean, you missed weeks and weeks of school because you were hospitalized, right? In
Yeah.
Bethlehem, Pennsylvania.
Hmm.
So, I mean, obviously I mean, the expense over a lifetime would have been quite extraordinary, right?
Yeah, I imagine.
Yeah.
I
Um,
mean, it's not obviously the expense of hospitalization is significant, but imagine the lost economic value for someone as talented as Marie. Um, she's now, college graduate, able to contribute very, very productively to our communities. That's priceless.
I'd like to turn to some of our viewers' questions. A number of our viewers are wondering whether CRISPR-Cas9 has application to other diseases. One of our viewers says that was wondering whether do you see CRISPR being used in type 1 diabetes, which is obviously a very, very common disease. Um, I'd love to pose to either of our physician guests whether that could be a treatment for type 1 diabetes or other diseases.
Yeah, I guess I'll weigh in first, and George can perhaps. I think, the one can imagine a myriad of applications of CRISPR-Cas9 to many disorders, certainly to rare inherited disorders where one knows the gene and the particular mutation. Those are particularly apt for CRISPR-Cas9. I think the issue for something like diabetes, I'm not an expert in that area, but you have to know what cells you want to edit. You have to know the gene you want to edit, precisely where you want to edit. And those are the things that we knew in this case that made it so favorable and opportune to use CRISPR-Cas9 on sickle cell disease. Uh, Not all diseases do we know exactly what cell or what gene or where to edit. And I think that's the problem when one thinks about diabetes or Alzheimer's or Huntington. You have to know what you want to do, and you also have to be able to get to those cells, the access to those cells, which is critical. And so there are many components that made this treatment for sickle cell disease, in a sense, a test case and a first application. and there'll be second, third generation therapies and other ways to deliver the editing components, but I think that's all for future research.
Dr. Daly?
Yeah, I think that Dr. Orkin's point is well taken. This disease was prime for cure by CRISPR-Cas9. It just was, and so it was very fortuitous. But this is going to be the first of, I think, a wave of genetic medicines. In some ways, this is the first opening of an era of genetic medicine. There are various forms of blindness, rare forms of blindness, where a similar kind of gene edit could prove very effective. HIV cells can be rendered resistant to HIV by a CRISPR gene edit. Other conditions like muscular dystrophy, cystic fibrosis, there are thousands of genetic conditions which potentially could be treatable by this strategy. But we're moving on to other challenges. Being able to deliver the gene editing machinery in vivo, in the body, as opposed to what Marie had, where the cells that we were targeting in her bone marrow had to be extracted from her body, put into the laboratory for the gene editing, and then given back to her with a transplant. That's a cumbersome and expensive procedure. What we'd like to be able to do is to learn how to encapsulate the gene editing machinery in a particle that we could just inject intravenously or intramuscularly that would home and travel to the target tissue and do gene editing within the patient's own body. That's the kind of technological advance that we would need to ensure that this therapy could be more widely accessible, and maybe over time the cost goes down. Because certainly if you want to scale even the existing casgevy gene therapy, you need a very, very sophisticated academic medical center that can perform bone marrow transplants. That's not going to scale across sub-Saharan Africa. So we continue, and I know Dr. Orkin continues to push the envelope of the science to try to make this therapy more widely accessible and easier to deliver.
So, so you're saying, it sounds like you're saying that even as remarkably as effective as this treatment, casgevy, is for sickle cell right now, this might not be the way that even sickle cell ultimately is cured. That there's going to have to be some new innovation along those lines. Is that Dr. Right? Is that right, Dr. Orkin?
Yes, I think that's right. This is the first generation. So it's remarkably effective, which I think is special for first generation treatment. There'll be second, third generation, I'm sure, and it's really progress of research that's contributing to this.
Marie, when we spoke a few months ago, you told me about how these pain crises would occur, and they often coincided with a change in weather, like the arrival of the cold. And you said that you had to take precautions, and now you don't. Can you talk a little about that?
Yeah, so I live in Pennsylvania where we have winter and snow. So when the seasons would change, I would feel my body start to freak out a little bit. Um, and now post-transplant, it's a little there's like a mental aspect to it because there's an anticipation for, oh, the weather was freezing rain yesterday. I should be feeling pain. And you kind of have to remind yourself that you're no longer experiencing this trauma that you've had for the 20 years that I've had sickle cell. So there's a big physical change, but also a really big mental aspect to the whole CRISPR treatment as well.
Right. You said, I think you said something along the lines to me like, wait, I don't have sickle cell anymore, so I don't have to worry about that. I
Hmm.
mean, that has got to be such a remarkable change in your life. I mean, um, I'm sure your family is flabbergasted by this.
Yeah, and it's something that we're still dealing with because my dad has sickle cell too. So he is still very jealous that he at this point in time can't get the treatment, but it's we were always in it together. So seeing me go outside without a jacket and he has to put 3 layers on, it's a very big discrepancy now. And it's like whiplash almost, and we're still getting used to it.
And now, and why can't he get the treatment? I mean, obviously you got it during a clinical trial, but has he tried to get this treatment commercially since it was approved?
Not since it was approved, but during the trial period, he unfortunately was too old, um, for the process. So I don't know if he'll qualify now, but I know given his age and what the process looks like now with chemo, if we can avoid him undergoing chemotherapy, that would probably be best for his health in the long run. So he may unfortunately be too old for the CRISPR therapy right now.
I'll put the question to either of the doctors. There are about 100,000 people in the United States who are estimated to have sickle cell disease. Is there a point when your body has too much damage that you can't get a treatment like this?
Well, you know, we're still gathering information about this type of treatment and its side effects, its long-term toxicity. Um, the bone marrow transplant itself does involve treatment with high-dose chemotherapy, and it has significant complications. So if a patient already has suffered sickle cell for many, many decades, and maybe already has a significant degree of organ dysfunction, that makes the transplant more dangerous, and more fraught with these side effects. So we need to learn more. We'd ultimately like to make the transplant less invasive. We'd like to be able to figure out how to deliver these gene-edited cells without having to use chemotherapy to prepare the patient. These are all technological advances that we hope will make the therapy safer.
Yeah, I would also add, I think the FDA approval is restricted to patients with severe, what's described as severe sickle cell disease, which is estimated, I believe, to be around 20 to 25,000 individuals rather than the 100,000 total. That may change over time, but that's the current stipulation.
Right. We're discussing the first ever approval of a CRISPR-based treatment, which happened in December for the blood disorder sickle cell disease. One of our viewers is asked a question about a disease that I believe, Dr. Daly, you mentioned, Huntington's disease. Now, and they're wondering whether CRISPR might be used to treat that. I would think Huntington's disease would lend itself to a treatment from CRISPR. I could be wrong, but I thought that was caused by one gene, and therefore it might lend itself to that. Is that the case, Dr. Daly?
Well, Huntington's is a single gene disorder that involves a sort of duplication of genetic elements within the Huntington's gene itself. It is a disorder that is inherited, but affects individuals later in life, oftentimes showing up in their 40s or 50s and leading to significant movement disorders, considered one of the neurodegenerative diseases. The challenge there is, when do you intervene and how can you successfully be able to target the gene editing to a distributed region of the brain with many, many cells already impacted. So as Dr. Orkin was making the point earlier, there are many, many different genetic diseases, but solving the challenge of delivery of this editing machinery is really, first and foremost, very, very difficult. So it makes certain conditions easier to approach than others. It doesn't mean that there aren't very exciting potential applications of CRISPR, for instance, in Huntington's disease research. I've actually collaborated with a group in Portugal that's used CRISPR-Cas9 to effectively revert the Huntington's gene in cells in tissue culture. So we're certainly learning using the technique, but for Huntington's, it may be a ways off for actual clinical applications.
One of the things that's so remarkable about CRISPR was, as I mentioned in the intro, it was first described really just about 11 or 12 years ago in a landmark paper by two future Nobel laureates. And to get a treatment developed within 10 years with this completely new approach, as I understand it, that's considered like warp speed, is it not?
Yes, I think so. It is warp speed. I think even more than that, uh, in our research, we were early adopters, I would say, of CRISPR, because it was obvious to, I think, all researchers that it was a revolution for science. We described in 2015 the precise region that casgevy edits. And I believe that the first patients who were in part of trials, probably received their treatment within 3 to 4 years of that. So the time frame is even shorter than you described. It's really quite remarkable.
One of the things that everyone's talked about is what an arduous process this is. I mean, you have to get chemotherapy to make room in your bone marrow for these transplanted, gene-edited cells. That it seems so complicated, and yet the doctors are talking about the notion of one day maybe delivering this as a pill. That's a little hard for me to wrap my head around, how you could convert something like that. Dr. Daly, can you break that down for us a little?
Whether I described briefly an alternative strategy to what we call ex vivo gene therapy, which is gene editing happening outside the body. We harvest the cells from the bone marrow or the blood. We put them in a petri dish and we do the gene therapy in the laboratory and then bring the cells back to the patient. One of the real advances we need is to be able to encapsulate the gene editing machinery and deliver it into the bloodstream so that it can go to the cells in the bone marrow and do the gene editing directly. A pill, in fact, is being sought for this type of therapy because if we could have a small molecule drug that you could take as a pill, that would, in a sense, reactivate the fetal hemoglobin that Dr. Orkin has spoken about. We could do it not genetically, but chemically. And that's actually very exciting. And it could, therefore, be an easier way to scale the therapy widely, let's say across sub-Saharan Africa, where transplants are few and far between. Now, of course, that would not be the same type of one and done genetic cure of the disease. You'd have to take the pill presumably for your entire life. But these are the kinds of strategies that scientists are continuing to work so that we do have next generation treatments. The treatment that Marie got is just a proof of principle. It's an exciting, dramatic proof of principle that it works. And now it's going to engender lots of follow-on science to make it easier, cheaper, and more widely accessible.
Dr. Orkin, I see you nodding your head. Do you feel that ultimately the current casgevy will be supplanted by much simpler and easier to use therapies that can be used all around the world as opposed to, places that have very, very sophisticated hospitals?
We would hope so. We would hope there would be easier to deliver therapies, whether they're in vivo therapy, as George described, with an injection or a pill. I think we, at this point, don't know which is more likely to be successful. But I think there'll also be other approaches to gene editing of the kind that the casgevy delivers, which could in fact be even more effective at increasing fetal hemoglobin. Although the clinical benefit, might be marginal if you're already at the 95%, hopefully cure level. So I think there's there's a lot in the future. We're just sort of at the beginning of this, hopefully a revolution in genetic medicine.
One of our viewers says, please talk about the ethical implications of gene editing. I assume she's referring to the whole notion that some people have expressed concern about for years of like using gene editing to change the basic building blocks of let's say, embryos to create designer babies. Is that a legitimate concern, Dr. Daly?
Yeah, so I've been part of 3 different international summits that have tackled this issue of the ethics of gene editing. And the most recent one in March of last year, held in the UK, talked a lot about the excitement around sickle cell treatment. But we were also left with the ethical quandary of broader access. And so, that is in and of itself a concern for bioethicists. But the issue that you could use this gene editing technology to actually change embryos is ethically contentious. Now, a couple of the folks have written in the question and answer asking whether when Marie is treated for her sickle cell, whether she could still pass on the gene genetically to her children. And indeed, since the therapy she received treats only her blood cells, indeed, her germ cells or eggs would continue to have the gene and could pass it on. But what you're talking about is, could we use gene editing to actually treat the embryos and cure sickle cell, even in the context of in vitro fertilization? Now, it's a very contentious issue. Some people feel that we shouldn't be gene editing embryos at all. Others feel that perhaps that might be the most efficient means of not just curing Marie, but making sure that her kids wouldn't have a risk. I was intrigued that Marie said her dad is impacted by sickle cell, and there was the known issue that she was at risk. So this is an issue that we debate in the scientific and biomedical community, and it does raise real ethical concerns. But I think as we get more and more comfortable that we can do this intervention safely, and that's another question that was put into the Q&A, we do believe that this is a remarkably targeted, molecularly precise gene edit. And yet we still have to follow individuals who've received the therapy, perhaps for decades, before we really learn the long-term safety. But we need to answer a lot of these questions before we ever even imagine going into something like revising the genes within an embryo.
Dr. Orkin, Dr. Daly was just talking about certain risks. Is there a risk of off-target editing when you do something like this treatment for sickle cell disease? Is that a risk?
Yes, this is a much discussed topic for editing, that there are potential off-target effects. That is, the Cas9 or other enzyme of that kind doesn't go to the right place. It goes somewhere else, maybe at a lower efficiency, and modifies a gene that one doesn't want to modify. it's always a risk. A lot of preclinical work was done in this setting to check for that, and it's really quite the guide RNA, as it's called, that guides the Cas9 to the right place for casgevy, is remarkably specific. In fact, many different experiments have been done to try to detect off targets in the laboratory, and it's remarkably specific. So I think one can never say it. There's no chance, but I think the chance has been reduced as much as one can in this kind of setting. And we'll have to see. That's, I think, another reason why the patients who are treated will have to be followed for a long time, perhaps for their lives. But hopefully there will be no side effects.
Yeah, if I'm not mistaken, aren't the patients going to be followed for like 15 years or something like that?
I believe it's 10 or 15 years, but I suspect that they'll want to be followed for their lives.
Marie, I don't want to embarrass you. I know that when I interviewed you, you told me how you missed more than 100 days of high school while you were hospitalized repeatedly for sickle cell. Yet somehow you managed to complete 8 advanced placement courses, play clarinet in the school band. You got admitted to BU. You did later transfer to Cornell University. We're not going to hold that against you. Um, and as I understand it, you have continued to do terrifically. You have gotten a new job even before you graduated from Cornell. Is that right?
Yeah, I'll be a management consultant with PwC, and I'm really excited.
That's Price Waterhouse Cooper.
Yeah.
Yeah.
Very good.
And it's amazing. It's absolutely amazing. I can imagine that that might not have been in the cards for you if you hadn't gotten this treatment.
Yeah, and even before the treatment, I want to make a note that I was an anomaly almost, that I was able to do as much as I did, that people with sickle cell are not able to um find full-time employment. They're not able to have a great quality of life just because of how often they have to miss out on things because of their symptoms. Um, so while I'm grateful for every opportunity I had, it was not without cost. And I know many fellow patients did not have that opportunity too.
Yeah, I remember interviewing a bunch of sickle cell patients, and I was struck by how many of them had recently started a new job or had lost a job because they had so many pain crises, and
Mhm.
they weren't able to work. I assume Dr. Daly and Dr. Orkin, you've seen that as well.
Yes.
Absolutely.
Yeah. Well, this is a subject that we could discuss a lot longer. It's absolutely a fascinating topic, and obviously, we'll all be watching to see whether this revolutionary treatment ends up being used for other disorders, maybe not even just inherited disorders. Um, I want to thank our guests, Marie Tournier-New, Dr. Stuart Orkin, and Dr. George Daly. And I want to urge our viewers to listen to more events on the Globe's Biotech Week. This is Jonathan Saltzman from the Boston Globe.